And we're almost done with chapter 10. And hopefully we'll get through a large chunk of 12 today, because that way we'll have time for the review on Friday before Monday's exam. Let me know if there's issues with the sign up like there was before. Okay. So we ended by talking about emerging viruses, and we just had a quick snippet of, coronavirus isn't something new. It's the new part of it is that it's global, a global pandemic that's more than 2 people. So a pandemic is classified as a pandemic if it crosses country lines. And these are arbitrary lines for a virus, especially in a global society. So for instance, Ebola was characterized as a pandemic. And that one, the one I'm talking about is even after this slide. So this is 1976, but the one I'm talking about was a few years ago, and you might have heard about it in the news, maybe. And it was classified as a pandemic because we did have transmission in Dallas. Okay, so, but that was like, I can list one person, right? So even though it's characterized as a pandemic, it's not comparable to the numbers that we see for going on right now, right? But that's a known issue when you have increasing pressure of society. And we'll learn about that in the last chapters when we talk about conservation biology, and how the closer you get to the natural habitat, the closer you are and able to get these zoonotic outbreaks. Okay. And the zoonotic outbreaks have the bigger potential for hitting a global pandemic because we're not prepared for them. Even though we had a SARS-1 in 2003, we didn't have a vaccine because, can anybody tell me why you didn't have a vaccine? It's not that we didn't try. By the time they got it to vaccine development, there was no one to run trials on because the outbreak had died out by that point. Because it had a higher case fatality rate, and you had a fever and you knew you had it and you couldn't transmit it without a fever. So they could track it and they could stop it, and people were more aware of it because it had a 10 to 13% case fatality rate, which is way higher, right? So by the time they got to vaccine development, that vaccine would have helped because it has very similar spike structure, but it unfortunately there was no patients by the time they got there. Okay, so and the example virus in your book, because your book predates obviously the coronavirus, is HIV as a retrovirus. It uses RNA as a template for making DNA. And what that means, anytime you see the word retro, you can have RNA without it being a retrovirus. Corona has RNA, but it's not a retrovirus, okay? What a retrovirus is a subtype of RNA virus that inserts its genome into your genome, okay, into its host genome. So HIV is a human retrovirus, so it inserts it into your genome. Because it has to insert, it has to make a DNA copy first so it matches your genome, and your genome gets tricked into integrating it. So since it gets tricked into integrating it, it has to go to DNA first. And if you remember the central dogma, now that we have a real biologist in here, I can't say the central dogma of all of biology. I have to call it the central dogma of molecular biology. But like I said, it's more important than anything else, right? So DNA makes RNA, makes protein. And this is called a retrovirus because it breaks this central dogma. But it can break the central dogma without breaking the actual effectiveness of it, because viruses are not alive. And we talked about that, right? So it goes backwards from RNA into DNA. And to do that, it can't hijack the cell's enzymes to do that. It has to bring its own machinery. And that particular piece of machinery is called a reverse transcriptase. If you notice when they talk about corona and they talk about testing, sometimes they don't just call it a PCR test, they call it an RT-PCR, depends on the source and how specific they're getting. But an RT is because they're artificially adding a reverse transcriptase from a mouse virus, because they have to convert it to DNA before they can test you, because we'll learn about the process of PCR in chapter 12, but you need a DNA template. Same thing here. Okay, so this is an example of what it looks like. This is its, you know, we talked about viruses have replication cycles, and the replication cycles are really what you want to target for drug because it's whatever looks different from us. And so the differences here on targets, attractive targets for drugging would be reverse transcriptase, for instance. There's also ones not mentioned here, but it involves in the process of putting it in. There's a specific RNA protein there. And then there's also a specific protein down here that involves its ability to go reinfect another cell. But those unique processes, you can see them on a replication chart, and those are what you want to target for drugs. Okay, so let's watch a quick video.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the
Sorry, that's my bad. I'm checking the chat while we're doing this.
Envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of 2 molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named, retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it...
So you know the process we talked about when we talked about cell division, it crosses over and the strands break and they cross over? That's what the virus is doing. It's tricking the cell. It's like, oh, you have a break. I have the homolog, not that other strand. Use this to fix it, and it gets into the genome that way.
May lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
So that process at the very end is random. And if we have time, I'll show you the randomness with a 3D printed model that I have in my office, but it's kind of loud, so we'll wait till the end in case it bothers people's ears with the speakers right here, okay? And we had an interesting question in the chat, and I couldn't respond to it in the chat during the video because that's when it paused, so I had to wait till the video was done. Jordan wants to know, yes?
So those viral cells that replicate inside the host cell, they leave and then they continue to infect other host cells.
Yes, yeah. Now, if you remember the first, so Dr. Bay, for those on the Zoom, he was asking if they will move to another cell and infect the next cell, and they will. And so there's different, if you go on to microbiology, you'll learn different replication cycles and modes of transfer to other cells. And so sometimes they're called latent, and they hang around for a long time, and that's not characterized by popping, the capsids popping and going to another cell. But when you get pain, when you have a lot of inflammation response, that's usually called a lytic cycle, where it's popping, it's budding from the cell and destroying the cell in the process, and your body has a lot of response to that. Okay, and we had an interesting question in the chat talking about all these different zoonotic viruses, and the question was, have we heard about COVID crossing over? And I want to clarify spilling over. So spilling over into minks, because she thought she heard something about a mink farm, and that's true. There were some mink farms decimated by this because corona is very promiscuous. The ACE2 receptor is pretty homologous across different species, and so we don't know exactly which species it crossed over from. It's the closest in nature that it's similar to, and the closest even closer than stuff in labs, in case you hit that theory somewhere, is a bat that was isolated in the wild. But for instance, SARS-1 came from a bat, but it was like a cat called a civet cat got infected from the bat guano, and that cat then transmitted it to humans. And so there's usually like an intermediary host that can, and you pick up a little piece, and we can see that in the genome. In those genes that it takes with it, when it buds out, we can analyze them and compare them to the different homologous viruses from those animals and track where they came from forensically, okay? So yes, it did spill over into minks. There's also been cases of proving that it has spilled over into some large animal cats at zoos. They can see that we were the intermediary host to those cats. Humans infected those cats at those zoos. And it's probably happening more often than that, but a domestic cat in your house is not going to have, like, you know, if they have some phlegm, they don't have like a large animal vet watching them all the time and worried and sequencing what's in their phlegm, right? So that's called spillover. When it spills into us, an interesting word for when it spills into other animals has been proposed, because this is not a phenomenon that we're scientifically used to. It's of course possible, but not scientifically used to it, and we call that spill back. Okay. And so one thing to mention is that now it's global, and even though we might want to blame China for their hygiene or something like that, you need to be aware that now it has the spillover potential for whatever is spilling back from us in this country. So one of the common colds, coronaviruses, in the 1900s, there was a record of a lot of deaths, and they thought it was the flu, but now they're thinking it might have been this common cold coronavirus, and it spilled over from cattle. Okay, so that's an example of this spillover phenomenon. Spill over and spill back, which is a new kind of idea. Okay, so that's the end of chapter 10. I left all these interest slides at the end because I want to encourage you to use them for study. Even though there's quite a few of them, I'm just going to flip through them so you can see the possibilities for when you study. Okay, including the checkpoint questions that we didn't get to in the learning catalytics, but we do have a learning catalytics coming up, so get your computers ready. Some videos we didn't play due to time. And these videos are not your publication videos, so you might want to check them out, just a different perspective of looking at the same kind of stuff we're covering, like replication. More practice, if you Google, remember we had all those clickers on mutations and the DNA code and the codon wheel, right? If you Google codon wheel worksheet, you will come up with tons of worksheets if you feel like you need more practice. Okay. Let's get into chapter 12. Chapter 12 is my favorite, which is going to make it hard to go at a normal pace. So we'll have a little bit of it on Friday, but it is a shortened chapter, so you don't have to worry about it being quite as large for what we need to cover. And your learning objectives, etc. Okay, so the introduction to this is to mention that Hawaii's papaya industry seemed doomed a few decades ago, because there is this deadly pathogen called the papaya ring spot virus that had spread throughout the islands. It appeared poised to completely decimate the plant population. Okay, biotechnology, the scientists at the University of Hawaii took interest, and to rescue the industry, they by creating a new genetically engineered version of the plant that was missing the receptor that the spike recognized. Okay, so this plant couldn't be infected by that virus. So today the papaya industry is once again vibrant, and the vast majority of Hawaii's papayas are GMOs, because they're missing what this virus can attack on the cell surface. Yeah, in the chat, I agree. Terminology is cool there. Okay. So in addition to GMOs in our diet, DNA technologies affect our lives in many other ways. We have this capability called gene cloning and editing that can produce medical and industrial products. DNA profiling has changed the field of forensic science. And then bioinformatics has become more attainable. For instance, I teach a non-biology class, just an honors class, and I taught them how to use Science Google and look up viruses and see where they came from. And it took a week. So it's become a lot more usable because everybody can use Google. And instead of putting a query like, where do I find Applebee's? Instead, you put in AGCCATAGGCT, and it says, oh, you can find that on the corner of Main Street and 12th. I actually don't remember the names of the streets, so that's inaccurate. But Science Google is a lot more accurate than that. And then what it does, just like Google, it's looking for a match. So bioinformatics as a field has popped up and informs a lot of the biotech. Okay, so some big ideas here: gene cloning and editing, genetically modified organisms, and DNA profiling. I want to encourage you, if you have questions about the ethics of any of these, to put them in the chat or bring them up, because we do want to talk about these. Anytime we have increases in science or technology, it's going to butt against the possibilities in society and create issues if scientists don't communicate the safety or the practice of them. Okay, so and by not talking about it, it just makes you concerned. So you might as well ask a question in a safe room. Okay, so gene cloning and editing. This will be our primer. So go ahead and open this. I'm going to deliver it. You don't have to know the answer yet because it's a primer. As soon as I find my, oh no, I closed my website, so I'll have to open it. And then we will get to the answer for it here in about 10 slides. But keep your eye out for it. So hopefully you're reading this slide while I'm doing this. I just started this session, and I'll write down the session ID if you need it. And then I just delivered. So I'm putting the session ID in the chat while people are logging in. Okay. So read through that. We already have a couple of responses that are already correct, which is good. Tells me that you're reading ahead, or maybe I've had it in high school and you remember it. It's not always the case that you would remember it, right? Okay, so gene cloning is one application of biotechnology, the manipulation of organisms or their components to make useful products. Researchers can manipulate what are called bacterial plasmids, and they can act as a molecular vector for us. So let me write that word down. These act as vectors. The word vector means it carries something. So what this is carrying, it's carrying our gene of interest. Sometimes, like literally you get into grad school, they use the word gene of interest so often they call it a GOI. And then if you haven't come across that term, you say, what's GOI? What gene is GOI? Because you know it's some kind of abbreviation, and then the professor, oh, that just means gene. You don't know that? Gene of interest. But in here, it's more about just seeing them as puzzle pieces that you're connecting together. So these, because you're connecting the vector that's a plasmid with your gene of interest, they're called recombinant DNA plasmids. And because it's a complete circle, so I'm going to do a complete circle, and then, you know, our gene of interest, I'm going to color code it a little bit different. So we keep in mind it's recombinant. It has something that's not native to it. The gene of interest, we slipped it in. It's going to be represented by this green bar. Because, however, the bacteria just sees a DNA circle, and if you remember, bacteria have circular chromosomes, then they think it's just part of their DNA structure, and they take it into their cell, and when it's time to replicate, it replicates. And so you have tons and tons of bacteria that have these plasmids, recombinant plasmids. In particular, we care that they still have that green bar in there and they have the gene of interest. So when they multiply, it's called a colony or a clone because they're replicating all the DNA within them, and they will replicate that plasmid. Now, it depends on what we want. Sometimes we want the DNA, so we destroy the bacteria and we use techniques to pull out the DNA. So we can harvest DNA if you want it. More often though, in the biotech world, what you're harvesting is a protein or an enzyme. Something not native to bacteria, otherwise we wouldn't be talking about this. Maybe sometimes a human gene that you were literally have a vat of the stinkiest growing thing ever, and you burst the cell and you pull out a human protein, and you have to ultra purify it because the toxins from the bacteria, but then you sell it on the shelf. So sometimes we want protein. And I'm going to put it in green because we really don't care about the bacteria. We care about the DNA that we put in there, that gene of interest. Or we care about the protein that's going to be made through the process of transcription and translation from that little green bar. This is what it looks like instead of spending a lot of time going step by step. What we're looking at is a figure. So you have this plasmid. That will be our vector, but it's not recombinant yet because you also need to go to your organism of interest and pull out that gene of interest. Should write it in green to be consistent, gene of interest. So where it's red right there, that's like what I was coloring green, okay? And then you're gonna recombine them. The recombination is because we have enzymes that cut at a particular DNA sequence. And if you look right here, the cut is indicated by this jaggy pattern that we'll talk about more. And then once the jaggy patterns match, just like Lego blocks, they match the right way. Okay, once they match, because they're using the complementary rules that we talked about in the previous chapter, you have your gene of interest recombined into that vector. It's recognized by the bacteria as, oh, that's something that I should also have inside me. Okay, so we have little holes poked into the bacteria, and the bacteria is like sucks up the nearby DNA. And then you have tons of bacteria if you grow it. Basically, it's not even like if you grow it in the right conditions. If you just like leave it, you know bacteria, right? If you look inside your refrigerator, if you've left something too long, it has grown, right? So really, this is a really robust technique. The undergrads in the lab do this a lot. This is one of the first things I train them on is grow bacteria and pull that DNA circle out. And then what you can get are either the protein or the DNA. Okay, so the genes can be used and inserted into other organisms. Sometimes you just want to propagate the DNA, or you can harvest the proteins directly that the bacteria is making, and you can use them as injectables, such as insulin is one example we'll talk about. Anti-clotting factors like antithrombin is another that's produced en masse. And there's a question in the chat about how much DNA you can put in a bacteria. There's features on the vector circles that you can design like a Frankenstein, because this is called genetic engineering. So literally you build it like a car part, and you can order different parts from the company that you want to put into your vector. And so if you want a high octane fuel or something, like that's the same thing with this vector. What you're going to choose is if you want a high copy number, you can choose that, and then the bacteria will make a lot of it. If you don't want a high copy number, you just break the origin of replication a little bit, and it doesn't do it as well, and it's a low copy number. So you can control how much the bacteria makes. That was a good question in the chat. Okay, zooming in, we're going to take that gene. The gene is going to have those sticky ends that are represented by the zigzaggy pattern. Now, very importantly, when those sticky ends are generated in your gene of interest, you also have to generate complementary sticky ends in your vector. They're going to then fit together the right way. And then you're going to use our friendly ligase that we learned about in the last chapter. They just purify it from any cell because ligase is pretty conserved. And you just put it into the reaction and it will suture up the backbone of the DNA. And then you have a circle that's ready to go into the bacteria. This is called transformation. You're going to transform that bacteria with the circle you just recombinantly engineered. And then as the bacteria reproduces, it also replicates and reproduces your DNA circles. And the genes can be inserted so you can purify the DNA, or you can directly get the proteins out. So some examples of some GMOs, because this is a GMO. If you put a non-E. coli gene in a circle and you put it in the E. coli, that's a genetically modified organism. Okay, so some examples of this are the glowy fish. If you go to Walmart, like you can go in Walmart right now and you can over lunch and you can see glowy fish. And what they did is they put a gene from jellyfish that fluoresces. And they put it into the genome of these fish. And so now these fish express that protein and they fluoresce different colors because there's different genes. There's a green fluorescent protein and there's a yellow fluorescent protein and there's pink and blue. There's all kinds of ones. And so they put it into that genome. Some other examples of GMOs, maybe a little bit more practical. So for instance, if you find an organism, Chernobyl, they looked, does anybody know what Chernobyl is? Okay, let's just say you find an organism growing somewhere it shouldn't, a really disgusting place that you didn't think life was possible, and you see something growing. That means it has genes that help it survive, probably by metabolizing whatever's gross. And so if you can find that gene and you can take it out, you can put it in something else that you know will say grow in the ocean, an organism that's okay with the ocean atmosphere. It has some protective things so the cell doesn't burst. You can put that gene in there and then you can inject your bacteria near that waste site and it will chew up or metabolize the waste. And so that's called bioremediation. Other examples of how you can use this is there's a gene found in, where did it come from? I know caterpillars can't eat it without crystals in their stomach. I think it came from a bacterium. But what they did is they took this gene that normally if the caterpillar eats this bacterium coated on something, then it develops crystals in the gut of the caterpillar and makes them sick. So what they did is and it doesn't harm other insects. It only harms this particular type that eat the plant. It also doesn't harm, so it doesn't harm beneficial insects like butterflies and bees. It doesn't hurt us at all. And we knew that extensively. So what they did, and that's important to know that we knew that extensively. And what they did is they took that gene that codes for the protein that makes those crystals that makes the caterpillar sick, and they put it into corn. And so this is GMO corn. Has this gene called the BT gene, and caterpillars can't eat it. Another example is human insulin gene is put into bacteria and produced. It has to be purified really well because you have to get it away from the bacteria. And there's also, like we mentioned, anti-clotting factors, proteins that are produced. Okay, so let's look at the process, and this will help answer your primer question, and I'll check the chat and answer it after we push play on this.
We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid.
Okay, so we've been representing sticky ends as just a shape. So let's talk more about what those are. So the restriction enzymes are what that cut into it. The enzymes that shown in the purple blob in the video, those are called restriction enzymes. And there's different types. The different types all represent enzymes that only recognize certain DNA sequences. These DNA sequences, interestingly, are usually palindromic, meaning if I put a G here, I'm going to put a C on the end. An A here means a T on this end. And maybe we'll do another A and another T. Oh, actually, that's your question for the learning catalytic, so let's change it. Okay. And if you bend it right here, using the base pairing rules, this would fold back on itself. G, T, C. And that's why they call it palindromic. They think this makes some kind of bubble in the DNA, and the restriction enzyme matches what it looks like when it folds back on itself in that way. Specific for what the sequence is, because it will make a little bit different bubble in the DNA depending on the sequence. Okay. And so what it's going to generate is usually indicated by a little a cut indicator. And that means, remember that DNA is double-stranded, and we have to follow the base pairing rules because this forms 3 hydrogen bonds there, 2 hydrogen bonds to the A, 3 hydrogen bonds to the C, 3 hydrogen bonds to that one across that base pair, A to the T, G to this C. Okay. And then the DNA goes off into the distance. But this particular site, the enzyme pauses that. And it says, okay, whoa, I match. Okay. And so it's going to cut, like molecular scissors, is going to cut through the backbone here. And if you read in the reverse direction, that's exactly the same sequence, right? We have a G first, and here we have a G. We have an A next. And then down at the bottom, we also have an A. We have a C, then we have a C. We have a G, then we have a G. We have a T, we have a T, we have a C, we have a C. Okay, so it also has a cut site here, and it also cuts there. So if you look, your pictures have shown a zigzag pattern because that's where it's going to cut and pull apart. The hydrogen bonds do tend to stick together though, and that's why it's called a sticky end. You will have an overlap. So at this one, once the reaction's done, if we look at the left-hand side, it's going to leave behind a T that's lacking a hydrogen bond partner, a G that has 3 empty hydrogen bonds, a C that has 3 empty hydrogen bonds, and an A. And that makes it sticky. It'll only match the sequence that matches it. And that's why you use the same enzyme for the vector as you do the gene of interest, because it generates these matching ends. Okay, this is a video. Well, let's go ahead and watch it. Sometimes I like to, if I feel like I've really explained it well, but you're always surprised by the questions that pop up when you present it a different way. So I'm going to give this video and let me know if you have questions about it. And yes, it's answer to the Zoom. That is in the BT corn. It is protecting against the gross little worm thingy that burrows into the ear of the corn.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts.
So what it's doing is showing you different enzymes. It says this one, it doesn't give you the names, but the names are named after the bacteria they found them in. Because the bacteria originally used this to chop up a virus. Something that's not native to itself, it's going to send that enzyme and it's going to chop up the virus. Okay, so they name it after what they purified it from. So common names are like EcoR1, HIN3, BAMH1, like weird abbreviations.
The DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA.
This is one we didn't talk about. Restriction enzymes don't always generate sticky ends. This is called a blunt cutter.
Sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
Okay, so that's really drilling down into it. The restriction site, you'll identify it on either side of your gene of interest. Use a matching restriction site. How do you know which restriction site will match? It's the bioinformatics thing I told you about. You just put it into Science Google, your sequence, and it will spit back out which restriction enzymes you need to purchase. The purchasing's not cheap, so they range from $60 to like $500, so it can get expensive. But those sticky ends generated, it's a pretty robust reaction. It'll either snap right back into place or it'll look for something else you've cut that way. So you do have to screen for the success of your reaction. You add the DNA ligase in and it will suture it back together and then it can go into your bacteria. So let's finish up the learning catalytics that we started that you should have been looking for the answer to, and let me see how you guys are doing. We're rapidly finishing up. It looks like we have 20% that still need to answer. Great, we're almost there. Okay, and we have about 50% correct, but there's a competing answer that is just the full restriction site itself, not the sticky end. So I'm going to open the slide back up, reteach that real fast, the distinction between sticky end versus the cut site, and then I'm going to close it, okay? So just to clarify, when I say something is the sticky end, or when anyone, like when your book, when Dr. Nesbitt, anyone that's knows how to genetic engineer, when they say sticky end, they mean the overlap. They don't mean the entire restriction site. So the restriction site will be clarified in that way. It'll say this is the restriction sequence that the enzyme recognizes, and it'll be the full thing. But the sticky end's what's left after the reaction. Okay, so I'm going to close if that helped some people. It did. That's great. Okay, I'm going to close it. And we're going to go to the second one. It's still a primer. We haven't hit the information for it, and it might be left open if we don't get to that information today. But I'll give you a second to read this because I want you to look for this information on the next few slides. And it is verbose, so I'll give it a second to read. Okay, so it'll be open. You can keep reading if you're still in the process of reading, but you should have a flow of what you're looking for in the next few slides. So we're going to talk about GMOs and how they're used. So in bacteria, yeast, human cell cultures, even whole animals, they can all be genetically modified using different delivery techniques. Same kind of engineering to get it in, but then how do you get that DNA into whatever you're talking about? Bacteria is the simplest, but as you get more complex, the delivery methods get more complex. And you have a problem with not all proteins work in everything. They all code because the DNA code is nearly universal. So human insulin can go in bacteria, huge jump, right? But you still can't use some human proteins because the bacterial system needs to modify it a certain way that it's not capable of for it to be a fully functioning protein. So examples of that are like, for instance, on the cell surface, the ABO blood grouping that we talked about, that's a sugar on the surface of your cells. And bacteria can't put those sugars on the proteins. So you couldn't generate, say, a protein that would mimic a blood type, for instance, because it doesn't have that sugar in the bacteria. So this is because bacteria were lower let's see, kingdoms, lower complexity organisms, even within the same kingdom, complexity of the organisms can't always modify the protein the way it needs done. So if insulin needed that sugar and you injected insulin made in bacteria into humans, then either the human would have a reaction to it because it doesn't look the right way, or it just wouldn't work. Luckily, insulin in our particular example doesn't have a sugar that's needed, so it can be made in bacteria. So there's some type of system-specific necessity, usually a modification. You can't cross organisms. It's not universal. The code is universal, not the modification. Some examples of where it is used, though, for instance, is this is a gene for a human blood protein that's secreted into the milk of goats. And so this is antithrombin clotting factor. They took the gene and they put it in the goat genome. And this is what's really cool. They put it behind the DNA sequence that says, hey, you're a milk protein. It's not, it's a human protein, but it says, hey, you're a milk protein. You need to be secreted into milk. And so when the protein folds, it gets secreted into the milk, and you don't have to, like, for instance, you have to kill all your bacteria to get the protein out. You don't, however, have to hurt this goat because he's producing she's producing the antithrombin factor, human antithrombin factor into the milk. It can either be purified or it depends on what protein it is. It can be consumed if it survives the digestion. So researchers use DNA technologies to produce drugs, diagnose diseases, produce vaccines, harmless variants or mutants of derivatives of pathogens. This is called forward genetics, when you make something that's to predict. This what would need to be done if you have a virus that you suspect could cause a disease in humans. You would have to make that virus ahead of time and study it in the lab. And so this is where you have biosafety level 3 or even biosafety level 4 labs would do this. An example, a friend of mine works on Plum Island and works with viruses that cause major economic damage if they got out. And so he has to take a ferry across, let's see, Plum Island's off the east coast of, I can't remember, we'll have to Google it. And you take a ferry across the channel, and he has to completely disrobe and leave his clothes in a separate room. He has to go into another room. He has to be sprayed down, and then he has to be, his anything that goes in and out has to be treated, because sometimes you do have to have things go in and out with UV light that would destroy anything. And then he gets to start work. So and then they have to leave the island by a certain time because the entire island is shut down. Okay, so when you do work with pathogens that aren't harmless, you have a lot of regulation on it. And how are these useful? So since ancient times, people have selectively bred agricultural crops. This isn't something new, but GMOs are new because you're adding a gene from a different organism. It's not that you bred a desired allele that already had that gene, but it was a different strain. Instead, you take a gene from something completely different and put it in. And how are they using it? So some differences in terms: genetically modified organisms contain one or more genes introduced into that genome by artificial means. But they can be a gene that you just took from a different strain. It could be, you're still modifying the genome, but it's not something different. And then a distinction for something different, at least one gene from another species, is called a transgene. And so that's like human insulin in bacteria. That's a transgene and a GMO at the same time. So transgenes are all GMOs, but not all GMOs are transgenes. And an example of this is in your book talked about is golden rice. It's a transgenic variety of rice created in 2000 with a few daffodil genes. And they're yellow because this gene is producing beta carotene that doesn't have a normal pathway in the golden rice, but the daffodil does with the yellow color. Okay, so but the beta carotene, if you consume it, is really good for humans because you can use it to make vitamin A. And so they made golden rice because it would be more nutritious. Your body could use it to make vitamin A. So they did the same thing, but their delivery method for plants, remember I said you have to have, in particular for different types of organisms, it's a different delivery method. They had a vector called the TI plasmid. That's what plants need instead of a bacterial plasmid. And this particular plasmid will flip it into the plant genome. And then you have a strain that will produce vitamin A. So genetically modified organisms are used in crop production because they are more nutritious and cheaper to produce. But the question is at a cost to potential bioethical issues. So for GMOs, scientists investigate the potential risks actively and continually before it's approved and after it's approved to monitor. But it does work. So golden rice is able to deliver vitamin A and is proven effective in delivering that vitamin and helping to solve the issue of vitamin A deficiency among children who rely on rice as a staple food. And to date, no study has documented health risk in humans from GMO foods. And it's not yet possible to see the long-term effects because we haven't been using it that long, but it's a desirable resource that we might have. So we'll leave there. I'll leave the learning catalytics open since it's verbose. That might be best so you can read it and work on it if you needed some more time to read. And we'll pick up with the rest of chapter 12 and then a review before Monday's exam next time. Okay, and I'm gonna get the virus in case anybody wants to see it. So it's all hydrogen bonds. It's always hydrogen bonds. And what it is these are magnets representing the hydrogen bonds, and the capsid is made individually in the cell. And then the cell is just like a little, and you can see that the virus starts to assemble. and it will pop from the cell just by collecting whatever it takes from the cell while it starts to assemble. And it glows in the dark. Not normal viruses though. One of the possibilities is partially true, but not for all organisms, just to mention about the learning catalytics. So consider the one that's true across the board for artificially GMO produced proteins. Okay, we'll end there.